Hello, this is Reward Life Science. Today I will demonstrate how the RWD Single Cell Suspension Dissociator can help you with tricky tissue dissociation with the least manual operation. For oncology, immunology, and neurology, the most physiologically relevant model may be primary cells that are isolated directly from tissue, rather than transformed cell lines. However, manual dissociation might lead to poor cell yield and cost a lot of time to repeat the meaningless procedure. RWD Single Cell Suspension Dissociator could improve the processing efficiency of tissue dissociation and guarantee the quality of cell samples, like the yield, viability, surface markers keeping, etc. There are two models for RWD Single Cell Suspension Dissociator. 4 channels device DSC400 and 8 channels device DSC800 could be used for high-throughput tissue processing. And for better dissociating performance, we have developed matching consumables, tissue processing tube. Based on the physiological characteristics and cellular composition of different tissues, we have also introduced the corresponding targeted reagents, the tissue enzymatic digestion kits. To start the tissue dissociation by RWD Single Cell Suspension Dissociator, you need to prepare the enzyme from RWD or your own protocols. We have various choices of kits for your applications, like tumor, brain, heart, liver, skin, adipose tissue, etc. All the kits from RWD have matching visual programs in the machine, so that you can just search for the name of the tissue and go with it. Or you can program your customized processes, because operators can set all the parameters, like the spinning speed, duration, and the intelligent software will make the programs compatible with more applications. Beyond that, 500 customized programs could be stored in the device and exported by USB port to synchronize with other devices. For more applications, we have considered the throughput of sample preparation too. Each channel of the RWD Single Cell Suspension Dissociator works independently, so you can dissociate 4 or 8 kinds of tissue at the same time. Usually, we should put the culture dishes in the water bath or warm shaker to keep the system temperature at 37 degrees Celsius when the enzymatic dissociating is taking on. A better way to do this is that you can put on the amazing accessories, so that you can select the built-in programs with the heating function or turn on the heating step of user-customized programs. The stable motor speed ranges from 0 to 4,000 RPM. So the single cell suspension dissociator is not only used for preparing primary cells but also homogenate. And both two kinds of sample preparation can be realized by the tissue processing tube. With the fully sealed design, independent packages, and EO sterilization, the tissue processing tube can prevent the biocontamination of harvested primary cells and keep them safe. Additionally, the single cell suspension dissociator also can keep the operator safe thanks to the in-position detection, which will send the alarm and stop spinning immediately when the tubes are detached. All you need is to prepare the tissue samples and enzyme reagents, mix them well, then choose the suitable program and run it. RWD Single Cell Suspension Dissociator will take care of the rest. After a simple filtration with a cell strainer, you will harvest the primary cell suspension for downstream experiments like single-cell sequencing, flow cytometry, cell culture, etc. If you have any further questions, please feel free to contact us.